Today I will cover the basic anatomy and physiology of eye movements, saccades and smooth pursuit. Conjugate eye movements are what bring visual stimuli onto the retina, allowing fixation on a moving target or to see stimuli in our periphery. The structures responsible for this are the cranial nerve nuclei and their extraocular muscles, but also controlling signals originating in the cerebral cortex, cerebellum, and vestibular structures. These can be divided into supranuclear, nuclear, internuclear, and infranuclear centres. The control of saccadic eye movements is from the supranuclear centres of the frontal eye fields and parietal eye fields, while smooth pursuit primarily involves the visual cortex. The eye in primary gaze is actually off the sagittal axis by 13 degrees, which affects all directions of eye movement except horizontal movements carried out by the agonist-antagonist pair of the medial rectus and lateral rectus. The medial rectus acts to adduct the eye with oculomotor innervation, while the lateral rectus abducts the eye with abducens innervation. Vertical gaze is more complex with use of the superior rectus and inferior oblique for elevation, and inferior rectus and superior oblique for depression. In detail, elevation when abducted, performed by superior rectus, is innervated by oculomotor nerve. Elevation while adducted is performed by the inferior oblique, innervated by the oculomotor nerve. Depression while abducted is performed by the inferior rectus, innervated by oculomotor nerve. And depression while adducted is performed by the superior oblique, innervated by the trochlear nerve. The conjugate eye movements can be termed dextroversion to the right, lavoversion to the left, supraversion or upgaze, infraversion or downgaze. Dextrocycloversion and lavocycloversion are movements which allow image stabilization and relative orientation for both eyes, whilst incyclovergence and excyclovergence affect the correspondence of retinal images turning inward or outward together. In horizontal gaze, the eyes must move together left or right, using the agonist-antagonist pair of the lateral rectus and medial rectus. For example, on moving gaze to the left eye must abduct with the contralateral eye simultaneously adducting. The connection between these is a white matter tract within the brainstem called the medial longitudinal fasciculus, or MLF. It connects the abducens nucleus for abduction to the contralateral oculomotor nucleus for adduction in horizontal gaze. For horizontal saccades, the control mechanism is at the frontal eye fields, which then acts on the paramedian pontine reticular formation, or PPRF. The PPRF is able to generate the saccade from excitatory burst neurons, and this is then conveyed to the abducens nucleus. The MLF is able to convey that signal to the contralateral oculomotor nucleus to control synchronized adduction of the contralateral eye. Vertical conjugate gaze and torsion are mediated by structures in the pretectum, with some cortical control from the frontal and supplementary eye fields. The rostral interstitial nucleus of the medial longitudinal fasciculus, or RIMLF for short, and the interstitial nucleus of Cajal, or INC, are highlighted here and are the most important structures in the control of vertical conjugate gaze. Upward and downward saccades are mediated by burst neurons within the paired paramedian RIMLF. Coordinated upward gaze functions through neurons from the RIMLF projecting to the ipsilateral oculomotor nucleus, before bifurcating, with some fibres crossing the midline to ensure bilateral innervation of the superior rectus and inferior oblique. The RIMLF also sends fibres to the interstitial nucleus of Cajal, which acts as a neural integrator, coordinating signals from both the RIMLF and vestibular projections. Each interstitial nucleus of Cajal has fibres that cross at the posterior commissure and ensure coordinated gaze. Similarly, for downward saccades, the RIMLF supplies the ipsilateral inferior rectus subnucleus and the ipsilateral trochlear nucleus, which affects the contralateral superior oblique. Again, signals are sent to the INC, which innervate the contralateral nuclei. Each RIMLF also contains neurons for ipsilateral torsional saccades. Internuclear ophthalmoplegia is a dysfunction of the adducting portion in lateral gaze. It is an injury to the internuclear neurons of the MLF, which can arise from vascular, demyelinating, or neoplastic origin. This damage can sever the connection between the abducens nucleus and its contralateral medial rectus subnucleus. In addition to this lack of adduction, on lateral gaze the abducting eye becomes dysmetric, there may be an upbeating or torsional nystagmus, and there is defective vertical smooth pursuit and defective vestibuloocular reflex. Examination for conjugate gaze dysfunction involves not only examining eye motility, but provoking response to smooth pursuit, head movement, and observation of saccadic function. This examination is thoroughly explored in Strupp et al. With the link below. In summary, horizontal, vertical, and torsional gaze is controlled by structures within the brainstem with some supranuclear modulation. This video has described the normal and pathological structure and function of the conjugate gaze centres using the references above.